So thank you for joining us for the 9th seminar in the Plant Phenome Journal monthly webinar series. My name is Seth Murray. I'm an associate professor at Texas A&M, also editor at the Plant Phenome Journal, and I will be moderating this webinar. So you can watch past webinars from the Plant Phenome Journal on our YouTube channel if you just search for Plant Phenome Journal. And you can learn about cutting-edge work going on in plant phenomics at the TPPJ website and the articles that have been published there. So today, Dr. Silvas Prince, and I don't want to butcher your last name, but Dr. Silvas, he recently had an article that was accepted and is now published with the Plant Phenome Journal. He's a researcher at the Noble Research Institute, focusing on root phenomics, and he's been there since 2016. Before that, before that, he was at the University of Missouri, where he was a postdoc from 2011 to 2016, and he did much of his previous work in India, and he's worked on, as we were discussing, a wide array of crops. So I think Silvas is going to tell us, Dr. Silvas is going to tell us more about his work, and please, let's welcome him to the Plant Phenome Journal webinar series. Silvas, it's all yours.
Thank you, Dr. Seth. I would like to thank the organizer, Dr. Seth, for giving me this wonderful opportunity to discuss about my research work on integrating phenome to genome to understand root system architecture in legumes crop. The crop production is very challenging as it is because it's very highly sensitive to different environmental fluctuations and climate changes. Crops tend to meet with the different abiotic stresses like drought stress, heat stress, flooding, and nutrition deficiencies and biotic stress, thus declining the grain yield as well as the nutritional quality of the food product. So the understanding of the root system architecture or the whole plant physiology is more important for researchers to enable to meet the human and the animal food needs and to deliver quality products for the consumer with higher nutrition quality parameters. The soil is an active ecosystem where you can find a lot of interactions between beneficial microbes and the plant. And the interaction between the plant roots and the symbiotic microbes are very important to maintain the soil health for a longer time to maintain sustainable agriculture. And the root traits are known to impact seed and biomass yield, and root traits are the target for the second green revolution, as Dr. Jonathan Lynch says. And finally, recently the root research gained momentum because of their highly impact on the performance of crop plants in limited soil, water, and nutrients. It's known that under drought stress, certain crop plants develop deeper and wider branch roots. But integration of the phenomics as well as the genomics part is very critical to make a big progress in integration of the root traits in the essential crop breeding that's undergoing in different crops. So on the left, you can see the difference in the rooting depth of soybean genotypes. Those are drought tolerant. For example, here the meningen 1 is a drought tolerant soybean. You can see it has a longer root length under water restricted condition. In the right, you can see the Kinadang Patong. It is a drought tolerant rice line that has a deeper root as well as the narrow root angle. So the scientists were very successful in introgressing a region that is governing the deeper rooting from Kinadang Patong, and then they introgressed through marker-assisted breeding approach, and they increased the root branching ability and the depth of the drought susceptible IR64. This is a very successful approach that has been proven in rice to improve the roots and which impact the grain yield under drought stress. In addition to the abiotic stresses, The root system plays a key role in accessing major nutrients. For example, the major nutrients nitrogen and phosphorus are needed for the plant growth and development throughout the growth stages. Under low phosphorus, the plants tend to reduce the total root length by reducing the lateral root density. And under low nitrogen, basically they tend to put forth longer axial roots where they can access the nitrogen at different soil horizons. The modeling approaches also showed that different roots has a different ability of nitrogen uptake, shown through the nitrogen uptake kinetic studies. In this study, they identified the lateral roots has the good ability to uptake nitrates, followed by the crown and the seminal roots. In this webinar, I want to emphasize the terminologies used widely in plant science community. The phenotype, it is the observable characteristics of the plant. For example, it may be a height, leaf size, and shoot biomass. And when you talk about the term genotype, it mainly describes the genetic makeup of the plant. When you see a phenotype out in the field, basically it is the interaction of the genotype and the environment. For example, to put it very clear, under drought stress in soybean, the soybean regulates different members of the aquaporin gene family. Because of this regulation, you can see the distinct phenotype in the field, like for canopy wilting, where you can see the leaves are wilted or they are away from the sun to withstand the drought condition. The roots are known to influence the plant health, productivity, and yield. But the introgression of the roots into the crop breeding program is very slow because of their inaccessible and there are a lot of it's a cumbersome process to identify some markers to introgress into the elite breeding program. So in my research, I explored the genetic diversity available in the wild crop relatives, where you can identify some new novel alleles to improve the cultivated crop varieties. In addition to the exploring the genetic diversity, we need to develop high-throughput phenotyping technologies that can enable to dissect the interaction between the genotype and the environment in a better way. So during my research, I addressed these three impacting factors that will translate the root into existing crop breeding programs. It's known that the seedling root, if you have a robust root system in the seedling stage, that translates to the later stages of the crop. So I established a phenotyping protocol where we can identify different root architectural traits in the seedling stage that will improve the yield at the later stage of the crop. So in here we grow this soybean in a cone for 14 days. After 14 days, the roots are harvested and analyzed using the wind rhizome, where it was quantified for different architectural traits. In the right, you can see the different parts of the root system of seedling legumes. Basically, the taproot is the root that emerges from the base of the stem. You can see here, and the lateral roots are the emerging from the taproots, and further branching of the lateral roots are known as the tertiary roots. Here in this presentation, I want to emphasize on the different approaches that we use to phenotypically characterize root system and to identify genomic region associated with the root growth using molecular mapping and GWAS approaches. The wild soybeans are known to be very diverse, genetically diverse, and they have a unique genomic content. So our approach is to identify the regions that are unique to wild soybean that can improve the cultivated soybean for stress tolerance and yield improvement. This is a mapping population developed in collaboration with the University of Nebraska. So the wild soybean parental line PI326 showed a higher root length than the max parent. And the progenies of this mapping population showed a transcript segregation pattern, and these progenies were quantified for different root architectural traits such as taproot length, total root length, surface area, average diameter, and root volume. Later, these phenotypic data were coupled with the genotypic data to identify genomic regions that are highly associated with different root traits. A significant genomic region on chromosome 8 was identified, and this region was found to control multiple root architectural traits like taproot length and lateral root number. And interestingly, this region is also known to associated with different yield-related traits such as seed weight, shoot dry weight, and the leaf internode length and protein content. The region from these two markers is around 138 MB, and it has around 500 genes in it. So, to understand the genes underlying these phenotypic variants, we screened those 500 genes, and we identified 11 genes that showed higher expression in roots and root-related tissues that can be further followed for other functional analysis. And this is another population that was developed with another wild soybean, PI 407162. And here also the progeny showed the transcription segregation pattern for different root architectural traits. In this study, we identified two key genomic regions, one on chromosome 6 that was contributed by the wild soybean, and one on chromosome 7 that was contributed by the cultivated soybean parental line. And we could see the increase in the tertiary root length based on the interaction between the region on chromosome 7 and the chromosome 15. And we selected some of the progenies with the wild soybean allele to explore further for their biological role in drought tolerance, and we tested them in field trials. We could see very clear difference when we have the progenies with the wild soybean allele. The root angle is much narrower than the wild soybean under drought stress condition, because the root angle is very critical in soybean under drought stress, because it increases the root length, as well as it makes a deeper roots where they can deplete water at deeper levels. And further follow-up experiment on the greenhouse, we could see the progenies with the wild soybean allele perform better than the cultivated soybean. And we did an assay for quantifying the root angle in both greenhouse and the field, and we could see the trend is similar across both the conditions. This is very critical when you have two different phenotyping platforms to integrate the data for developing a crop cultivars. The root morphology is known to influence different stress tolerance. And recently, the anatomical traits also playing a major role on the stress adaptation as well as the nutrient uptake. So similar changes were observed in soybean as well in our studies. And a key study that was done in maize by Jonathan Lynch group, they selected some contrasting maize recombinant inbred lines, contrasting for 3 different anatomical traits, such as root cortical parenchyma, cortical cell file number, and cortical cell size. The root cortical parenchyma is the air space that you can see in the left side, that those air cells are formed constitutively or under certain stress levels. So when they tested these contrasting maize lines, they identified when the maize lines have high RCA and the reduced cortical cell file number and larger cortical cell size, they tend to have deeper roots and explore like higher surface area with less root respirations. In our study, we used the nested association mapping panel. The nested association mapping panel was developed in collaboration between multiple universities and USDA. And the nested association mapping has both elite as well as exotic germplasm in soybean. Here you can see extensive phenotypic variation of the root anatomy in the NAM panel. And in the right, you can see the blow-up of the root anatomy, where the area highlighted in the red shows the parenchyma, and the area highlighted in the green shows the Meta-xylem number. The role of Meta-xylem is known to influence both water and nutrient uptake in croplands. We tested this NAM panel for reproductive stage stress in a rainout shelter at University of Missouri, and we identified there is a huge difference in the pod load or the pod number in this diverse set. And down below you can see the graph. Some of the lines has a higher pod number under stress, but most of the line has a reduction in the pod load under water limitation. The increase in yield or the decrease in yield was coupled with two different physiological parameters. The high yielding was associated with the lower stomatal conductance as well as the reduction in leaf area. If you have a smaller leaf area and if the genotype can maintain the stomatal conductance, that is more important for maintaining the photosynthetic machinery under stress. So we selected 3 contrasting high yielding and 3 low yielding lines from those NAM panel to do a mechanistic study to understand the role of anatomy in stress adaptation. And the common thing that we identified in the high yielding lines, they tend to have higher Meta-xylem number as well as the reduction in the Meta-xylem diameter. These two traits are very critical when you think about the water uptake from the limited availability. So we see the trend in these high yielding lines. They tend to have higher number under drought stress, and the higher number in the Meta-xylem also increases the hydraulic conductivity in the shoot in the high yielding lines in the greenhouse study. So further, we want to study the effect of the difference in maturity groups and the geographical origin on root anatomy and architectural traits. So this study was mainly aimed at understanding the effect of climatic adaptation of different soybeans originated from different places of the world. So here we tested 4 different maturity groups that was selected from different parts of the globe to represent a wider geographical distribution, and we selected 251 accessions that has been genotyped with the 50K SNP data. So when we did the clustering analysis, we identified 4 clusters. Basically, the 3 clusters represent 3 different center of origin like Chinese, Japanese, and South Korean accessions. And the 4th cluster is an admixture that has a combination of other 3 clusters. These 251 accessions showed a tremendous phenotypic variation for different root architectural traits. But the extent of phenotypic variation was higher for the lateral root number. So when we couple the phenotype and the genotype data, we could identify a high signal on chromosome 16 for lateral root number. So when we mined the region, we identified that is associated with the cell wall related gene. The interesting thing is that the variant that we identified on chromosome 16 is present only in 52. So it is a rare variant that is present in very few accessions out of total 19,000 USD accessions. And further mining, we identified These are widely spread out in late maturity as well as the early maturity soybean varieties. Here in the lower right, you can see this is a normal rectangular shaped cells, cortex cells in the wild type. So if you have this variant, the genotypes will show a square shaped cortex cells. So this is very critical when you have the surrounding cortex cells. That will decide the number of lateral roots, initiation and development process. Basically, that lateral root has to develop from the pericycle, and it has to push through all the way through the cortex cells to come out as a lateral roots. In addition to the 50K SNP data available, there are a lot of other resources that came up after that based on the whole genome sequencing or resequencing of soybean genotypes. With those data sets, we could identify additional variants in the same gene. So we want to explore further their effect of the additional variants in the root anatomy. So here this is a wild type that I said it has a rectangular shaped cells. But when you have these synonymous mutation that changes the amino acid content, that still changes the cortex cell shape to columnar shaped. So these changes in the morphology of the cell are coupled with changes in the phytohormones. We could see there is a drastic change in the auxin and ABA levels that are well known for their role in lateral root initiation and development through cell wall modeling techniques. So we took some of the accessions with those interesting haplotypes and we tested them in the field. So we want to see whether the response that we are seeing in the greenhouse are replicable in the field condition. We identified certain accessions. They showed higher fibrous root score. Basically, the fibrous root score is a visual score that will be done in the field based on the amount of branching from 1 through 5. And interestingly, you can see some of the accessions showed higher plot yield that is on par with the wild William 82, that is a reference genotype that are widely used. It's a elite cultivar. So this is an opportunity where these accessions can be used to improve the plant performance as well as the yield under drought stress. And some of the accessions showed higher protein content of 55 and 48.3. In soybean, the relation between yield and protein content is always inversely proportional. So these accessions can be used to improve the elite cultivars for both yield and nutrition quality, and to further study these complex traits such as yield and protein content and their relationship between them. The root studies in the field, that is a shallow mix, is a destructive approach, and they are highly influenced by the soil heterogeneity. Basically, it may be a water or nutrient or the soil texture. There are a lot of other factors that affect the accuracy of the phenotyping. But the lab studies are well controlled, but they never exemplify the field scenarios. So there is a need in the scientific community to develop a system where you can capture the field root response using a lab assay. So this is a good example of interdisciplinary success story that happened at University of Missouri. So we collaborated with the engineering department at MU, and we thought about thinking. We thought about making a platform that can be affordable, because the commercially available systems are like 15 to 20 grants. So we want to make a system that will be widely adaptable and affordable to use. So we developed a laser imaging platform to identify certain seedling root feature in the lab that can accurately predict the root rates that are measured in the field, such as root angle and fibrous root score. So here I want to explain the workflow of the system that we developed. The soybean seeds are surface sterilized, and then they're germinated for 3 to 5 days in a sterile polycarbonate vessel. And after 5 days, they are transplanted into the artificial gel media. And the plants are grown in the artificial gel media for 14 days. And after 14 days, they are imaged using the laser light scanning system. The plants with the artificial gel media are kept in the turntable, and range images are generated. And after the range images are collected at different view angles, these are compiled together to construct a 3D imaging of the roots. So these are the components of the imaging system. We have laser points, and we have a Firefly HD camera, and this is the linear slide platform with the turntable that has a motor attached on the top. So the bottom picture shows the difference in the root branching ability of two different genotypes selected in this study. So for every scan, 360 images are made, and basically at every turn the turntable makes at this end, it makes a turn of like 10 degrees to change the view angle to capture the root image. The root images are reconstructed based on the deflections of the laser points. Here you can see the 3D constructed root image of a particular genotype, from where 11 different 3D root features are extracted. Here I showed a particular feature that's known as a convex hull, where it exemplifies the total vertical length of the root as well as the extent of the root branching of a genotype. So to validate this imaging approach, we selected 9 different genotypes. Those are genetically diverse, and these 9 genotypes are tested in the field, and we have data on the root angle and fibrous root score for 2 years in 2 different conditions. And below is the visual fibrous root score visual scoring method, where the least branched roots are ranked as 1, and the roots with more branches are termed as 5. So we had 11 different root features, but we identified a 3D feature volume that is highly associated with the fibrous root score that we observe in the field condition. So when you select the seedling assay for a higher root volume, it might translate to the higher fibrous root score in the field condition. Here I want to talk a little bit about the phenotypic approaches that are suited to perennial alfalfa crop. The alfalfa different populations were tested in the field under 2 different management practices. One is the water limitation, and other one is the planting density. So the sward planting is the approach where the plants are closely planted each other, and in the bed 1 and 3 we can see the effect of the planting density, and in the bed 2 and 3 we could see the effect of water stress on above and below ground response in alfalfa. For this study, we selected different alfalfa population contrasting for root types. So here you can see the branched alfalfa population we selected, we had 2, and for the tabbed roots phenotype we had 2 populations. And in this field study we used high throughput phenomic strategies to study alfalfa response over the cropping season. And we developed indices like NDVI that resembles that tells you about the healthiness of the plants. And we developed some script based biomass estimation. And the data collected through the scripts were compared with the actual biomass collected throughout the cropping season. In the raised bed experiment, the alfalfa population with the branched roots, that is Mariner 4 and the U30 to 33, showed higher yield under both well water and drought stress. So they had higher biomass throughout multiple cuttings. We did 5 cuttings, and then in all the 5 cuttings, the branched roots performed better than the tabbed and other individuals. In this study, we identified the effect of plant spacing and the water availability on below ground response. The population FSG 420 is a tabbed population, whereas the Mariner is the branched population. Under sward planting, both the populations showed reduction in the root branching ability. This is due to the intercompetition between plants, the nearby plants, and the competitiveness for soil and water nutrients in the available plants. So the closed planting and the competition between interplants reduces the efficiency of root system as a whole. Here you can see these factors reduce the ability of plants to produce thicker and highly branched roots. The crowding stress that happened in the sward planting had higher intraspecific competition for solar, radiation, water and soil nutrients. But under spaced planting, the branched as well as the tabbed tend to have more branches under drought in contrast to the well-watered conditions. The branched population UM30 to 33 tend to have higher branches irrespective of the water availability. So these are the different genomic resources and phenomic variability that we can use for improving the field persistence in alfalfa. Excuse me, one of the slide is not showing up, please. The roots were excavated after the cropping season to understand the below ground response in alfalfa. Here we compared 4 different populations contrasting for rooting types. The different parameters were studied. Those are root number, root length, width to depth ratio. This determines the root distribution with depth. The convex area that represents the extent of the roots that they grow to cover the soil area. The average radius denotes the thickness of the roots, and the shallowness that denotes the amount of root that is available in the topsoil for foraging the nutrients. On comparison of these 4 populations, we identified the Mariner 4 with the branched root type. They had higher root number and root length, and the root distribution is much higher in both the above soil as well as and in the depth. But the Bulldog 805 has a higher root system that is evident from its higher average radius data. And the imaging of the roots from the bed 2 and 3 with spaced planting method revealed that the branched Human 30 to 33 that is shown in pink had higher root number as well as the root length in the rain-fed condition. And this was followed by the Mariner 4, that is also a branched population that had higher root number as well as the root length. Both the branched population Human 30 to 33 and the Mariner 4 had higher width to depth ratio. And this distribution along the root might be the reason for their higher biomass under drought. The two populations, bulldog 505 and 805, they differ only for the fall dormancy. The fall dormancy is similar to the maturity group difference in soybean. So here we tested the two different cultivars that are differing only for the maturity group, and we identified they didn't show any difference in the root parameters between rain-fed and irrigated condition. The root traits influence only the water uptake and nutrient uptake, but the water movement within the plant system and the water use efficiency rely solely depend on the leaf morphology and its physiological processes. The two populations, PI 478 and PI 502, are originated from different climatic regions. The PI 478 is from a mild desert climate, and the PI 502 is from a cold and prolonged winters. On visual observation, you can see the PI 478 has a smaller leaf area in comparison to the PI 502 with a larger leaf area, and the check cultivars also had a bigger leaf area in contrast to the PI 478. And the three leaflets were individually analyzed using the leaflet analyzer software that was developed at Noble Research Institute, and they could clearly distinguish the three leaflets for different leaf morphological traits. But in different morphological traits, the PI 478 had a smaller leaf morphological attributes than the other two populations evaluated in both well-watered and water stress conditions. The complete root system were excavated from these three populations from the PVC and analyzed using in-house MATLAB script that were developed at computing services at Noble. The three populations differed for the root structure and the distribution along the different length of the soil horizon. During the processing, we compartmentalized each root system into 4 different sections to get more details on the root distribution in top, middle, and the bottom soil layers. After segmentation, data on root angle, root area, and the total length was collected. The PI 478, 573 had higher root biomass under the bottom soil, but in the other two populations, the root mass were distributed along the PVC columns. In comparing the traits, we identified the PI 502 had higher root volume, and on regression analysis, the root volume is highly associated with the shoot fresh weight that were measured after the drought in the greenhouse study. This robust root system in alfalfa will enable better capture of water and nutrients, and they do cover a larger volume of soil to facilitate the acquisition and the use of nutrients. And this is an interesting picture I want to share with the community to show the importance of the root plasticity and the different stresses. Here, all these genotypes are the tolerant genotypes for that respective abiotic stress, but they show the plastic response where they tend to maintain higher root mass and under drought and salinity stress. And this is a peculiar response under flooding stress. This is The tolerant plant tend to produce more aerial roots above ground to access oxygen under hypoxic conditions. This response was observed in both greenhouse as well as in field conditions. And the root system is a key tool for the plant research community to reach the optimal grain yield under both marginal land as well as the highly nutritious lands. And to achieve the future agriculture in an eco-friendly manner, we need to combine the genomics and the high-throughput phenomics and the breeding applications to introduce root traits into breeding programs where it can be practically applicable in agriculture. And thank you for giving me the opportunity, and I would be happy to take any questions from the viewers. Thank you.
Well, thank you, Dr. Silvas. What a enlightening and wide-reaching seminar covering a lot of aspects of legume root growth. So we're going to open it up for the next 15, 20 minutes for questions. There's two ways you can submit your questions. One is you could type it in the chat section if you want. You can either address it to Dr. Silvas or to myself or just to everybody, and I will read it. Or you can take advantage when there's a break in the conversation to jump in and just unmute yourself and ask your question. Okay. So while you're getting your questions, I'll just make a brief plug for the Plant Phenome Journal, which is what Dr. Silvas's paper is in regarding some of this technology that he presented today. And it's an open access society-led journal that's really dedicated to advancing these issues. The other thing I'll point out is that there's a number of plant phenomics meetings too that are opening these days, and you should consider presenting your work and sharing those with the world in that way. So I'm going to start with a couple questions here. Oh, one last plug. Next month's seminar will be Dr. Larry Purcell and his graduate student and postdoctoral colleagues from the University of Arkansas, also talking about legumes and roots. So, Silvas, question for you. I'm not much of a root connoisseur. I haven't looked at nearly as many as you have. But when I'm talking to somebody like Alex Buckish at UGA, he showed me some amazing variation that he saw in the field with roots. And I know most of your work is not in the field, but have you noticed large variation within a genotype just due to field heterogeneity?
Yes, we observed the same kind of response when we tested wide diverse lines in Delta Center. That is the southmost part of the extension center that we had at University of Missouri. We could see some variation because they had a sandy soil, and where the soil, as you progress through the field, the end of the plot has higher clay content, and they have phytophthora as well, and they have I think it is like a root drought where they have SCN problem. So when you have this kind of heterogeneity in the soil, we try to replicate the experimental plot we had in the field, but still it's an unavoidable situation where you have to have an assay from the greenhouse. Basically what we did, when we see an extent phenotypic variation in the field, we try to follow up with the evaluation experiment at greenhouse. Basically we want to make sure that whatever the phenotype that we are seeing is for the trait of interest for a particular stress. For example, in my case, I'm really looking at drought stress. So I will do a couple of experiments with those genotypes that were screened from the field to make sure that we are capturing the phenotypic variation for that particular stress, and it is not confined by or maybe like affected by the other factors that are in the soil in the field sites.
So I think what you're saying is you try and control for it as much as possible, but it's almost impossible in the field setting.
Yes. Yes.
Have you or any of your colleagues looked at the percent variation in some of the traits, like root branching, for instance, that come from the field variation as opposed from the genetics?
We did see some variation for the root angle, but we could see the fibrous root score. Basically, we do a visual score from 1 through 5 in the field. So when you take that same set of genotypes for the branching intensity, it holds good than the root angle in a better way, because the rate of root branching that you observe in the field is much higher than the greenhouse, but the trend remains the same. But for the root angle, there is huge variation between different experimental setup when you have a PVC column or when you have a bigger pot in comparison to the field. So there are certain traits that you can rely on and proceed further when you see the exact phenotype out in the plot. But it's always, we do multiple trials and multiple locations. Basically, you want to try to do compare different soil types as well as the environment. So we worked with two different states. One is the Arkansas and Kansas, where we have wide range of variation in the drought stress as well as the soil edaphic factors. So we try to minimize the variation that we are seeing at a particular site rather than doing a wide scanning of for representing different locations to identify lines that can be used for breeding program. That will be, at the end, it will be widely applicable to different farming situations and the environment.
Okay. So try and control for it and average it out. We do have our first question from Dr. Daniel Lescavar. So he's wondering, is the 3D root imaging laser system commercially available, or is it still under development?
The system that we developed is basically, the parts are available, and I shared the components that are needed for this platform. It is easily accessible, and it is very, very cost effective to do that rather than buying a commercial system. But this is like a handy system where you can develop on your own, and we have the scripts available, and it is also published along with the paper that people can refer, and then the people can do on their own. This system is easily accessible, and it can be built on any places where you want to, and you can use it.
That's great. Any other questions? If you're asking a question, please unmute your microphone. Okay, while we're waiting, I guess I have another question for you, Dr. Silvas. So if you're talking about your SNP for lateral branching being rare, I think you said 52 out of 19,000 accessions had that SNP. I guess I'm a little concerned or confused. Is it possible that it was just such a rare allele that really inflated the effect size and the estimate of significance? Or did I misunderstand something?
So would you please rephrase the question for me, please?
Yeah, so if the SNP is that rare, 52 out of 19,000 accessions, then in your detection and discovery of the SNP on your panel of, I think, 300 individuals, is it possible that the effect size and the significance is inflated because maybe it was only found in 5 accessions in your panel?
No, because the thing is that, like, when we do the compressed mixed lineage models, we do correct the false discovery rates for by using different algorithms like Bonferroni corrections and stuff like that. But when you have highly significant SNPs under compressed mixed lineage model, it is a reliable SNP. And when I check the value of the phenotypic variation that can explain, it is above 10%. So that is a large effect SNP that can influence the trait. That's why we want to further explore the different SNPs in the same gene using the whole genome sequence, and to identify why we are missing this SNP in the whole genome sequence. Basically, we use the two different approach for the whole genome sequence itself. The 50K is a reduction where they use a restriction enzyme to cut the whole genome, the sequence to do sequencing approach. But in the whole genome sequence, you will have more number of SNPs because they use a different strategy where they can get the complete information of the whole genome itself. So we hypothesize that might be the reason that we are missing the other two SNPs that should be available in this 300 germplasm itself. So, and we could see those SNPs explain a higher phenotypic variation among the accessions only if they are, let me rephrase it. So the SNP that we identified here in this 50K SNP, the germplasm has only land races and accessions. Those are the plant introductions. But the other two SNPs that we identified are using the NAM and the allele lines dataset. So what we hypothesize is that the rare allele is present in the wild accessions and the local the plant introductions, but they are not transferred to the allele lines. So even when we check the sequence information, when the genotype has this allele, they won't have this allele. When they have this allele, they won't have the second one. But there are certain genotypes has the combination of the first one and the second one, and the first one and the third one. So we are thinking about the recombination and the evolution aspects as well. But when you have a combination of these two, the effect is less rather than having this alone. So there are a lot of other work needs to be done, and we did overexpression analysis, but I didn't have time to show all those results what we got. But we did the overexpression of this allele in soybean, and we showed the same response that changes the root cortex cell shape. So that is a confirmatory stuff that we did, but if we need more additional work to double confirm what's happening and what this gene is doing in the cell modeling techniques. So, but it has been proven that in Arabidopsis, if you have a square cell or a thinner cell wall, the lateral root primordial elongation from the pericycle is easier than if you have a rigid cell wall like this. So it has to break through all the cell walls to come out and make a lateral root. So there are a lot of other work needs to be done to answer your question like in a straight way, but this is what we got, and we think that this has a tremendous effect on the cell wall morphology that will regulate the lateral initiation and development process.
Okay. Well, I'm not seeing any other additional questions. Dr. Silvas, can you please check in your chat window, make sure nobody addressed them directly to you?
No, I don't see anything on my chat as well.
Okay. Well, thank you for an excellent seminar. If no more questions, going once, going twice. Well, if no more questions, thank you very much. Great job. And we'll look forward to seeing everybody next month.
Thank you.
Have a good day.
Thank you. Thank you for having me. Thank you.
Thank you.